So the goal of transcriptomics is to compare gene expression levels between different cell types or tissues within the same organism, or between, for example, healthy and diseased cells or tissues, and also between cells or tissues which were either or not exposed to stress conditions like a drug. So typically exposure to certain toxic compounds. Now we can do that because if you have a look at this diagram, DNA is transcribed into messenger RNA or mRNA, and this is then translated into a protein, and the protein will then carry out a biological function in a cell. So when we perform transcriptomics analysis, as the name implies, we are focusing on the level of the messenger RNA or the transcripts. So when you want to compare or determine gene expression levels, you could use different methods. The most common ones are real-time PCR. It's also called Quantitative PCR or qPCR. There is northern blot, then there are microarrays, and finally there are RNA sequencing techniques. The technique that you use is going to be decided for you based on your actual research question. Does that make sense? So if you want to measure the expression of one specific gene or a small set of genes, or whether you would like to perform a broader screening and measure the expression of thousands of genes and potentially even all the genes that are expressed in the genome of your organism of interest, that would dictate which technique you're going to use, which kind of makes sense. So techniques like real-time PCR and northern blot focus on just like one single gene of interest, whereas microarrays measure the expression of thousands of genes. And then you come to RNA sequencing, you can even measure the expression of all genes that are expressed in a certain organism. But as you can appreciate, with your expansion of data that you can collect, so not just one gene of a handful of genes, if you want to go to thousands, or if you want to then do the whole, then that is accompanied by costs, usually higher costs. So that's how these techniques track. Does that make sense? Okay, so let's look at some deeper details about each of these. If we start with northern blot, the first step is to isolate the RNA from your samples. After the RNA isolation, for northern blot, you will load the RNA onto an agarose gel and perform gel electrophoresis. This, of course, will allow you to separate the RNA based on size. Following that, the RNA will be blotted, as we call it, northern blot. So this means that RNA is transferred to an RNA binding membrane. And then lastly, you have to hybridize this RNA with radioactively labeled nucleic acid probes. And these probes you have to design beforehand. For it to work, the probes will have to be complementary to the RNA sequences of interest, the RNA sequences you're trying to target. And if you're familiar with electrophoresis in the protein setting, it's very similar, because here you will wash the RNA to remove any unbound probe, and then the membrane will be exposed to an X-ray film, and this generates what we call an autoradiogram. And this here, you can see this will look, and based on the intensity of the band that you obtain, this tells you something about the expression of your gene of interest. Are we on the same page so far? Let me know in the comments. The great thing about a northern blot is that it will give you information about the size of the RNA transcript that you're looking at. And this could be interesting to detect alternative splicing, for example. And also it will tell you whether your RNA product is degraded or not, because this is something you will also be able to see from the band. Another advantage of this northern blot technique is the specificity. Because you designed the probe, because there's a target you're looking for, if it does bind, then you can trust it. But can you think of the disadvantage of this technique? If you can, please let me know in the comments. So it's relatively low sensitivity. If we compare it to something like the real-time PCR or the sequencing that we'll be discovering, the whole genome sequencing. What this means in practice is that if the gene is low abundance, you're not going to necessarily be able to pick it up. You may get the wrong impression that it's not there. Another disadvantage is that the probe that you have to use is radioactive. That's what allows you to be able to detect it. And so, of course, there are some safety precautions. And because of these reasons, in recent times, people just would rather not. Because there are other options, safer options. If there weren't, then yes, you do it. But if you have other options, why would you, right? So the northern blot, like qPCR, is for when you're looking at only one gene or a small set of predefined genes. However, when you want to perform a broader screening, for example, when you want to test which processes are affected by a certain drug, a certain compound, you can use different methods, and one of those is the microarray. Have you heard of microarrays before? The main principle of microarray is, like, for example, let's take a simple scenario where we just have two samples that we're trying to compare, and we can label one as blue and the other one as green. Or this could be representative of a sample that's derived from cancer cells and the other are cultured from healthy cells. So just like in the northern blot, you will isolate messenger RNA from these cells. You will reverse transcribe it into cDNA, which stands for complementary DNA. So that's a new step that you didn't have in the northern blot step necessarily. So one of the cell lines, say the healthy ones, will be labeled with one color, let's say a green fluorescent probe, and then the one from the cancer cell line would be labeled with a red fluorescent probe. And then the idea of hybridization comes into it again, because now what you do is hybridize the cDNA to a chip that has predetermined genes or probes in them already, but in this case you can have hundreds to thousands of genes embedded on the chip, so that when your sample is added to it, they can hybridize to multiples of genes instead of just the one in northern blot or just a handful in your qPCR. So these sequences are complementary. Once again, that's how these principles always work. Whether it's in the northern blot or in the microchip setting, your probes are complementary to the target genes you're trying to detect. After the hybridization, you're going to detect if it's happened. So you scan these. Usually they have laser scanners. You shine a particular light onto the fluorescent probes, and they emit a particular wavelength, which is captured by the equipment, the instrument. And the fluorescence detected corresponds to a particular gene. And then the readout is made accordingly. If there's no emittance of fluorescence, then it means that gene was not in the sample. Does that make sense so far? Are we on the same page? So you can see that this method offers you the advantage of being able to measure 20 to 50,000 genes, for instance. This is something you wouldn't be able to do with real-time PCR. Well, you could, but it would be very expensive and also time-consuming. But can you think of any disadvantage with this technique? If you can, let me know in the comments. So the first disadvantage is that it requires quite a large amount of RNA. So I guess you've got 20,000 genes that you're probing. You need a lot of RNA. Again, you need to put the probes on the chip to detect it in your sample. And so you need to know the sequences you're looking for beforehand. So it's predetermined, predefined genes that you're going to detect. If you're exploring and wants to know what else is impacted, this will not be the right tool, the right technique. Does that make sense? Are we on the same page? So another disadvantage, if you are aware that there are other techniques, is that, like if you wanted to compare it to RNA sequencing, then you would say this technique has lower sensitivity. And this can be particularly annoying because if there are small differences between two different conditions, like the drug-treated and the non-drug treated, if the difference between them is small, microchip may not allow you to, a microarray may not have the sensitivity to pick that up. That real-time PCR, which tends to only give you one gene or a handful of genes, the workflow of real-time PCR is similar at the foundation. So you have to isolate the RNA from your samples, then you will remove any residual genomic DNA. You will have to convert your RNA into cDNA. Once you have cDNA, you'll be able to form the actual qPCR. Are we on the same page? Once you have the cDNA, before you can do the qPCR, you need to design primers. I have a video on here that will help you if you've never had to design primers to get an idea of what that process looks like. Would that be useful to you? You can check it out. You need to determine the efficiency of the primers once you design them. Is it clear to you why you need to determine the efficiency of the primer? If it is, let me know in the comments. And so once you have these primers that you trust, you can now use it to detect or measure the expression of the gene that you are interested in. And this process, you would have to perform data analysis. You have to normalize the data. Nowadays, the preferred technique for detecting differences in genes tends to be RNA sequencing. So just like the other methods, you will have to isolate the RNA. That's standard. But there's something different here where you have to fragment the RNA into short segments, and then you convert these segments into cDNA. Do you know about why you have to fragment the RNA before you make the cDNA versus when we were using the northern blot, or if you're trying to do qPCR, why in qPCR you never have to fragment the RNA? If you do, let me know in the comments. So after fragmenting and converting to cDNA, you will ligate these with sequencing adapters, then amplify them, and then you will perform next generation sequencing. So what happens when you do this is that from the next generation sequencing machine, you will get these things that are called reads, and you will then map or align these to a reference transcriptome or genome of the organism that you're working with. So here's the main point: how you determine gene expression levels between different treatments or between different cells or between different conditions. What happens is, depending on the number of reads that map to a specific gene, you then determine the expression of that specific gene in a sample and compare it between different samples. Can you think of the advantage of this approach where you basically just fragmented all the RNA, read it, then aligned it back to the transcription, what the initial expressed genome would have looked like? Can you think of the advantage? There are several, and what I can think of is that one of the first ones is that all transcripts that are expressed in your organism are analyzed. The RNA sequencing platforms have superior sensitivity and a large dynamic range, which means that it's able to detect very small quantities of transcripts, but also very large quantities of transcripts. And what makes this tool or this technique desirable is that you're not limited to known sequences. So the reason, of course, is that you're not working with probes that are complementary to your genes of interest, but you're simply sequencing all transcripts that are present in your sample. And what's really cool about this approach is that you've captured the transcriptome and maybe the function of the genes are not yet known. But then later on, a couple of years, several years down the track, someone explains, does experiments that explains what the genes are doing. You can go back to your data set and look at its expression in your experiments. Does that make sense? Another advantage of this is that it also allows you to detect a splicing variant, so you can also check whether exposure to a certain compound affects the expression of specific splicing variant of a certain gene. Do you know what the impact of splicing variants mean functionally? If you do, let me know in the comments. So just like before, where as you get more data, it's more expensive, the same thing happens here, because now you're not just focused on, depending on the size of the transcriptome, you may be looking at a lot of genes. And also because you're just sequencing everything, you're getting more data, it tends to be relatively expensive. But can you think of a disadvantage? So in the previous techniques, the gene, it's hybridized in the form of it's in a probe, and then you hybridize to it. So when you detect it, you know already what you're looking at. But in this setting, you don't know what you're looking at. You're exploring. So the data analysis is not straightforward. It's rather complex. You need good bioinformatic knowledge. Does that make sense? So these are the techniques that you use if you want to study the transcriptome. I hope that it was helpful, and if it was, please let me know which part was most useful to you. And I'd love to know what gene you are working on, which technique is going to be suitable for you. Maybe you're looking at a range of genes, or you actually want to do the entire genome. It's been fun sharing this information with you, and I'll catch you in the next video. Bye. God bless.